Okay, everybody, we are back for the second half of chapter 11. And so this word action potential we introduced in the first part. And so we should know now that an action potential, we see it abbreviated AP, but it really means to us a nerve impulse and a nerve signal that is sending away from the cell body down the axon. So here's another example of three A's. Action potentials propagate away down the axon. Okay, our electric wiring of the body is our nervous system, and we actually can measure our nerves in millivolts. So it really is like electricity, and most of our body is at an electric neutral. When we do have a charge, that charge occurs because we have an imbalance or a difference in ions on one side of a membrane versus the other. So when we're talking about our membrane in this example, this is zoomed in on our neuron. So if we put a probe into the cell membrane of our neuron, we can measure the internal environment versus the external environment. Potential energy is going to be measured in millivolts. And when we talk about our nerves, this -70 number is where our neurons rest during resting membrane potential. And so our neurons at rest are negative. When we send our action potential, we'll see it's going to become positive, and then we will return to that negative number. And that's part of what makes our nerves excitable, is being able to maintain this resting membrane potential. We said things like to go from higher concentration to lower concentration in diffusion. So that concept is going to be important for this. And in this general concept are greater difference in charges. The bigger the difference between the inside and the outside, the larger that membrane potential is going to be. So we're going to talk specifically about potassium, which is K plus, and sodium, which is Na plus. So these are ions because they have a charge. The plasma membrane is another way to say our cell membrane, and we just said that there's a charge because we have a difference in ion concentrations. We learned about the sodium potassium pump way back in Unit 1 because it was an example of active transport, which is why we see this ATP to ADP arrow here. And our sodium potassium pump, no surprise, is in charge of pumping sodium and potassium. But they're going to go opposite directions. And we need energy to do this because we're pumping them against their concentration gradient. So if we're going with the concentration gradient or down the concentration gradient, that doesn't take energy. We can think of that like we're paddling downstream, we're paddling with the current. If we want to paddle upstream or go against the concentration gradient, it's going to take energy in the form of ATP to do so. So our sodium potassium pump is going to pump sodium out and potassium in. And there's a reason we have these three and these two, because each time our sodium potassium pump runs, we're going to pump three sodiums out and two potassiums in. And this is how we're going to reach that -70 number, or our resting membrane potential. We'll also see we have different types of protein channels that allow entry and exit of potassium and sodium when they're open. And so this is going to be swimming upstream in terms of our sodium potassium pump. We need energy. We'll see when it comes to our channels, that's when we're going to swim downstream or go down the concentration gradient. So there's no energy involved in this part of the movement. So this is what our axon will look like at resting membrane potential. Sometimes you may see this abbreviated as RMP. And as we said, the two main ions that we're going to talk about are sodium and potassium. And so each of these are a positive ion, but don't let that confuse you because the inner environment here is going to be negative. Our outer extracellular environment is going to be positive. So when we're starting at resting membrane potential, all of our voltage gated channels are closed. When we say voltage gated, what we mean is that these are going to open and close in response to changes in voltage. So when we start off, we have a lot of sodium on the outside. We have a lot of potassium on the inside because our sodium potassium pump has pumped out the sodium and pumped in the potassium. So as we go through this, we want to kind of keep track of what our charge is like inside. That's what we're always referring to when we're looking at our graph. So we're about at negative 70 millivolts inside. We have a lot of potassium on the inside. We have a lot of sodium on the outside. And protein channels tend to be specific. So we have protein channels for sodium and protein channels for potassium. Speaking of our channels just a little bit more, we have a few different types of channels we're going to talk about. Leak channels are always open, so they're always leaky, and they're responsible for the permeability aspect of our semi-permeable plasma membrane. In this case, we're referring specifically to ion leak channels. We have a few different versions of gated channels. And so gated channels are going to open and close in response to a specific signal. Ligand gated ion channels were an example that we saw within skeletal muscle. So when our presynaptic terminal released acetylcholine into the synaptic cleft, that acetylcholine bound to a ligand gated sodium channel on the sarcolemma. We're going to primarily talk about voltage gated ion channels, meaning those that are going to open and close in response to a small voltage change. And when we say small, we mean we're talking in millivolts. So even though we're going from negative 70 up to about positive 30, overall, that's a relatively small change. Mechanically gated ion channels mean they open and close in response to mechanical force. This is something we'll see in chapter 14 when we talk about some of our receptors, namely our stretch, pressure, vibration receptors. So kind of just keep that one in the back of your mind. So we've mentioned this already, but now you're seeing it in words. Our electrical signal produced by the nervous system is an action potential. So nerve signal, nerve impulse, action potential, all of those are saying the same thing a different way. Action potential is our official name for it. We'll see that abbreviated AP. And remember, action potentials travel away from the axon. Our neurons obviously communicate with each other and other parts of the body. They're the only ones that can send action potentials. Our glial cells that we talked about in the first half, they're supportive cells. They're super important for our nerve function, but they don't have the capability of sending an action potential. Action potentials are where our neurons communicate with any effector, and that could be another neuron, that could be a muscle, that could be a gland. It's whatever is going to be our postsynaptic membrane or what our neurotransmitters from the presynaptic terminals are going to communicate with. So we said membrane potential is that difference in ions that establishes a charge. And so that unequal distribution creates a difference in that intercellular, intracellular environment versus our extracellular environment. We're going to really keep track of sodium and potassium. We'll introduce chloride at the very end of our lecture today. So we said this when we first showed our picture there, but we have a high concentration of sodium there on the outside we have a high concentration of potassium on the inside. So there's a big concentration gradient difference between sodium and potassium. So think in your heads now, when our voltage gated channels open for one or the other, which direction are things going to want to go? So kind of make your prediction and then we'll walk through it together. Like we said, a lot of our intracellular and extracellular fluid are electrically neutral, but in terms of our neurons, in terms of our membrane and resting membrane potential, we have an uneven distribution of anions and cations. So just introducing those as vocab words. I always remember the difference here because the T in cation looks like a plus sign. So our cations are our positive ions. Our anions are our negative ions. In this case, we're going to have an unequal distribution of two different cations that are responsible primarily for this neuron sending its action potential. And so we've already said this a couple times, but here's the word version. And if you're still struggling to understand this, there's a link to a video here, and table 11.3 in your book is also very helpful in understanding this relationship. I'll point out now, here's another link to a video to help you walk through this in a different way. And so we're going to walk through not just what happens within our cell membrane and what happens with these ions, but also how we can graph that out. So we have our charge here on the side, and we have our time here on the x-axis. So charge on the y, time on the x, and all of these different vocab words are going to be important to you. So we'll walk through all of these words together. Okay, so depolarization is when the inside of our cell starts to become more positive. So positive ions are coming in. In this case, we started with sodium on the outside. So sodium is going to start to enter our cell, and we're going to come up towards negative 55 millivolts. So we could draw a dotted line across this graph that we call the threshold, and we'll talk about what happens if we meet or don't meet this threshold. So the all or none principle is related to our threshold and neurons in general. So if we reach threshold, if our graded local potential is a strong enough impulse to reach the threshold, we hit that negative 55. As soon as we get to negative 54, there's no stopping the action potential. It's going to go all the way up to positive 30, positive 35. It really depends on what you're looking at. And so this is really what we refer to as depolarization. Sodium channels open, and so we have a whole bunch of sodium on the outside. And so once those channels open, we said our channels are going to swim downstream. They're going with the concentration gradient. So we have a ton of sodium rushing in. Sodium is a positive ion. So as it enters the axon, as it crosses the cell membrane, we're going to go from a negative environment up to a positive environment. So sodium is entering through voltage-gated sodium channels because it was this change in charge or reaching threshold that caused them to open. If this initial graded potential was too weak to reach this negative 55 number, say we only got to negative 60, then we have the none half of our all or none principle. We're not going to depolarize. We're going to return to that resting membrane potential. Repolarization is part B of our graph here. And so when we get to this peak, our sodium channels are all the way closed and our potassium channels are going to open. Remember, we started with a whole bunch of potassium inside the cell. We're still swimming downstream. So we're going from high concentration to low, meaning potassium is going to rush out. It's again a positive ion. It's a cation. So we're going to go from a positive environment back down to negative. Our voltage gated potassium channels stay open a bit longer than they need to, and so we actually get part C of our graph, which we call hyperpolarization, meaning we actually become more negative than our resting membrane potential. And we'll talk about this a bit more with our refractory period, but we do that so that we prevent sending too many signals too quickly. And then it's our sodium potassium pump's job to reestablish our resting membrane potential. So in a sense, once we have A and B and C happen, our sodium and potassium ions have swapped spots, and we now need to switch them again. We need potassium to get pumped in, sodium to get pumped out, and we can reestablish our resting membrane potential. I would star this picture here because this is something that you may see again. And so not only should we understand what each letter or what each piece of our graph says, but what is actually happening at the cell membrane to do this. So as a practice activity, see if you can put these in order. And I would say go ahead and pause your video for now and restart once you think you have your order down, because we'll walk through this together. Okay, so our resting membrane potential is at A here, or this line. The very first step would be our trigger zone on the neuron receives a stimulus. So really, our sodium potassium pump at this point is shut down because once resting membrane potential is established, it doesn't really have a job to do. The trigger zone of our neuron, which we refer to as the axon hillock, or the very start of our axon, is going to first receive the stimulus. So this initial curve upwards towards the threshold is going to be our graded potential or local potential. And this is really more controlled by those leak channels that are always open. If that graded potential reaches our dotted line G, that's our threshold, we know based on the all or none principle that we're going to get a full depolarization. So B here would be depolarization. And so we should know that means our sodium gates, our sodium voltage gated channels to be specific are now open. And so sodium rushes into our axon. We now have an action potential sent. The second half of our graph here is more of a reset. And so at the top here, our sodium channels close and our potassium channels open. So once potassium gates open, we're going to have potassium rush in, and they stay open longer than we even need them to. So we're going to go even farther down. So C would be repolarization, D would be hyperpolarization. And then think in your head, what is in charge of reestablishing resting membrane potential? And that's going to be our sodium potassium pump. We need to return all the ions to their original position. So not only should we understand what are our different labels, but what's actually happening. Now, you may be wondering, what are F and E referring to? Because we haven't really talked about that yet. F is referring to our absolute refractory period. E is referring to our relative refractory period. And so in F, we absolutely cannot send another action potential. So a refractory period means it's not sending a signal. So when we're already fully depolarized and we're working on repolarization, we can't send another signal at the same time. So there's absolutely 0% chance that that happens. E we called the relative refractory period because it's relatively difficult to send another action potential. And it's relatively difficult because instead of -70, we're down here at around -90. So it's going to take a much stronger stronger local potential or graded potential to get us to threshold. It's not impossible, it's just relatively more difficult or more unlikely than at resting membrane potential. So section F here is associated with the absolute refractory period. We absolutely will not depolarize again. Section E is with the relative. It's relatively difficult to depolarize again. For those of you that don't like seeing it in the graph version as much, here's a table version. So this may help you understand or organize your thoughts when we code through the action potential. So here's our difference between our graded potential and action potential that we just talked about. So the graded potential really starts within our dendrites and cell bodies, and that's going to be kind of the start of our action potential. So it's going to initially spread a very short distance and die out. It's just the very, very beginning. This is much more localized than an action potential, and it typically is more associated with our ligand and mechanically gated channels. Our graded potentials also tend to be proportional to the stimulus, meaning if it's a strong stimulus, we get a strong local potential. If it's a weak stimulus, we get a weak graded or local potential. And then action potential goes with all of those things that we just talked about. Okay, this is kind of what I was just talking you through in terms of F and E. So if you like seeing the words along with it, this may help. And so our sodium gated channels are inactive during this absolute refractory period. So if our sodium gates are inactive, there's absolutely 0% chance we're sending those signals. Our relative refractory period, we can still possibly get an action potential. So our potassium gates are still open. If we get enough of that impulse to reopen sodium and not as much potassium comes in, there's still a relative chance we reach this threshold. We mentioned this back when we saw our myelinated versus unmyelinated axons within the first part of chapter 11. So think again, which one goes with myelinated versus unmyelinated? So if you said saltatory goes with myelinated, you would be correct because we remember we have these nodes of Ranvier. And so the action potential really kind of happens at one spot and then jumps to the next spot, jumps to the next spot, jumps to the next spot. So that saltatory propagation means it's jumping over those nodes. It's not necessarily that our entire axon becomes positive at one time and changes back to negative. So it really happens in segments. For continuous, we don't have those nodes because it's kind of just pressed into the myelin sheath. It's not completely covered. So we can call that continuous propagation. There's no nodes to jump over. And I should point out that even though we see Schwann cell in this example, meaning we have a peripheral nerve, those nodes of Ranvier are present in both the peripheral nervous system and central nervous system. It would just be an oligodendrocyte forming the myelin sheath in our central system. And so this just gives you a little bit more or a different way to see that same information. I always like to point out this very bottom segment of this table because it seems like if we didn't have to jump nodes, it would be more efficient. But our unmyelinated axons are less efficient and slower, meaning they actually use more ATP to accomplish this task. Our saltatory conduction, our myelinated neurons, those jumps over the nodes of Ranvier, use less ATP and they occur much faster. All right, hopefully this is semi a review for you in terms of a synapse in these different terms here, because we talked about this in specific, kind of a specific context with our neuromuscular junction. And so we use that as a little bit of an example over here on this part of our slide. And so a synapse itself just means a junction or connection. This can be between two neurons. This may be between a neuron and a muscle. This may be between a neuron and a gland. So a synapse is our connection. The presynaptic neuron is going to be that initial neuron that's propagating the action potential. That action potential reaches our presynaptic terminal, our presynaptic knobs, the synaptic knobs. You'll see a few different terms for that. Those synaptic knobs, if you remember, are going to contain synaptic vesicles, are bubbles with neurotransmitters. Neurotransmitters are the chemical messenger of the nervous system. Those neurotransmitters are going to be released out into the synaptic cleft. And so that's our space between our pre and post-synaptic neurons in this case. And our post-synaptic neuron or post-synaptic membrane will be what receives that signal via the neurotransmitter. Some examples of neurotransmitters, acetylcholine is the one that we saw with skeletal muscle. Some other examples of this that you may have heard of are dopamine, serotonin. Sodium can act as a neurotransmitter. Calcium can act as a neurotransmitter. So there's a lot of examples of these. And for the most part, a neurotransmitter either says keep the signal going or turn the signal down. So we have a couple different types of synapses based on the type of signal. So for electrical, our action potential is going to travel through a gap junction. So the best example of this is our cardiac muscle. We have those intercalated discs that are our specialized gap junctions, and this allows really fast communication and synchronization so that our heart muscle is not beating all in random spots. It's all beating together. Our chemical, which we talked about with neurotransmitters, occurs across the synaptic cleft. So this is more of a chemical example, and this is a bit slower. And we can name this based on where we're going. So axon to dendrite, axon to cell body, or axon to axon. So we just said this, a neurotransmitter is the chemical messenger of our nervous system. All right, some of these images may look a little funny to you, but I think this is a great way to remember our two types of summation. So for you math people, when we add two numbers together, we call it a sum. So for summation, we're talking about the addition or amplification of a nerve signal. So we see here we kind of have two types. So for spatial, we have little Jimmy here is getting jumped by all of these different guys. And so all of these different guys get one punch in. So we have multiple presynaptic neurons giving an action potential to one postsynaptic neuron. So everyone in our green shirts are the presynaptic neurons, and they're all sending a signal to our postsynaptic neuron. For temporal, we have one presynaptic neuron that's sending multiple action potentials, multiple signals to our postsynaptic neuron. So now at least we have a fair fight. We have one green shirt, one blue shirt, but our green shirt is punching our friend over and over and over again, which is going to amplify the effect. So I think these are a great way to remember the difference between spatial and temporal. All right, very last thing to cover here, guys. Thanks for sticking with me. So, excitatory versus inhibitory. This is why I was saying that neurotransmitters really, no matter what we have, result in one of two things. They're either going to be excitatory or inhibitory. So, for those that are excitatory, it's going to help with depolarization of our postsynaptic potential. And so in this case, we're talking about a graded potential to be specific. And so we call that an EPSP. So EPSP, really just remember that E is for excitatory. And so this is going to cause sodium channels to open. And what that means is that our postsynaptic cell is going to get closer to that threshold. It's improving the chance of depolarization. The opposite is inhibitory. We can also call that hyperpolarizing because that's going to take us farther away from the threshold. And so we call that an IPSP. So this is going to either be a ligand-gated chloride channel or a ligand-gated potassium channel. And you see here, potassium is an anion, chloride is an anion, potassium is a cation. So they have different directions that result in hyperpolarization. So remember in our graph, hyperpolarized meant we went farther negative. So knowing what hyperpolarization means should help you remember which direction we're going to go. So if we're going to be inhibitory, if we're going to go farther away from the threshold, we need to go farther negative. So for chlorine, we're going to have a negative or an anion, meaning that's going to come into the axon to be inhibitory. So that's going to drive our potential farther negative, farther away from the threshold. We can have that same result by pushing more calcium out. So if we have more positives, more cations going out, we're still going to get more negative. So either way, we're getting farther away from that threshold. We're getting more hyperpolarized. All right, great job. Thanks for sticking around. I know that was a lot, but it'll help you a lot as we go through our next part of the unit.